So a couple of things came up in that discussion, which I also was going to sort of start off with or things that up until now hadn't come up. So the question was, what about that Bruce Dobkin thing? So for those of you who are not spinal cord researchers as your primary role, the spinal cord injury locomotion trial was a multi-center trial looking at the type of therapist-assisted locomotor walking that I saw at Shepherd yesterday, so I know you know about that, to actually examine what was supposed to be a slam dunk, which is that training will induce plasticity in the spinal cord. People who are trained on the treadmill will obviously walk so much better. They were doing it in the subacute, reasonably subacute population, which was seen as this is just insane to do this trial because everybody already knows it works. Which, as you well know, because of your involvement in all of it, it did work. It worked fabulously. It was a negative trial. Why? Because other kinds of therapies induced equally as rigorously, but not specifically treadmill training, did so much better than expected. Okay, so it gets back to the natural history question that we just talked about. And it gets to this question, was it a trial worth doing? Was it a successful trial or a failed trial? So the primary outcome measure of how well they walked was no different between the control group and the experimental group. But both groups, by any standard, did so much better than was already expected that in isolation, either of those would have been an absolute success in terms of functional walking. And it gets to something Wise said about with cleaning out the spinal cord lesion area, 54% of those people walked. And the question is, what would they have done in Bruce's trial without any manipulation of the spinal cord itself, which bore risks, but showed that in those cases they were not starting with diagnosed Asia A's. I realize that. But were they really Asia A's? There's lots of questions that were raised in between the lines of what we heard today that get at this question of how do we as a group decide when it's ready, when something is ready to take forward to trials. So,
Naomi, can I ask a question about that? What if what if the FDA isn't involved in what you're doing?
So a good example of the FDA not being involved is something like the Staskis trial, which is going on in Canada right now, a multi-center trial looking at early decompression injury. It's basically a surgical trial. It's a prospective look at a surgical technique, a clinical technique. Why? Because there could be no agreement. The surgeons for years and years could not agree on the ethics of not doing the surgery they felt was best for their patient, and the question of is early decompression justified or an unjustified risk to a patient has never really been rigorously looked at. So within the surgical community, without any interference whatsoever from the FDA or the equivalent in Canada, these issues come up. So don't put it all on the backs of the FDA either. So another thing that nobody else mentioned very much of is the issue of pain. Pain is a serious clinical issue which could very easily be a target of a clinical trial, but more often we think of it as the potential negative outcome which would put people at great risk for participating in a trial if instead of function they get back pain. And there have been some reports of that in people who've gone overseas to some of these clinical treatments. I don't know about China, but I do know that the Portuguese experience has had some reports of people going into pain after that. So I'm going to just not bring up any more. I just want to emphasize those points I didn't think came up.
So one big barrier is clinical agreement.
Clinical agreement. The ethics seen by the clinical community of going forward, and I would say also the basic community in bringing something forward to clinicians that it should be tested, which is back to, I think I may be too focused on the barrier I came in hand with, which is how much evidence do we need, but I'm going to turn that around and I'm going to ask Wise, what's the point of basic science?
What's the point of basic science?
In spinal cord injury. And why do you need it? I mean, you talked about feasibility, you talked about logistics as a legitimate reason for going forward, more than which cell is best or how that cell works or what the risks of that cell are or the combination.
Well, I find it so interesting that I get asked this question because I've done But I want to say that I have done basic science my whole life long, and I did not present any of the data behind the lithium and the umbilical cord blood, and I think it's not, that wasn't my intent. If we do want to discuss that, I would be very, very happy to discuss it. There's a huge amount of science behind this thing. There is, and I think it's just that we haven't discussed it. I didn't present it, it was a short discussion. But on the other hand, I think it's very important to understand that clinical trials serve not just to show that something works.
I agree.
It shows that something does not work as well. And this is an issue that just simply hasn't come up for discussion, I think, enough. So that I think basic scientists need to understand that clinicians, evidence-based medicine has multiple purposes other than to show that basic science works in human. It is to actually optimize clinical practice, to optimize clinical use of therapies. And this is, I think, a very important function of clinical trials. We don't recognize this enough, I think, in the field. So I want to say basic science is absolutely important. And look, every drug that we actually bring to clinical trials has to be based on preclinical.
So the question then is, how do we, as a group, look at the barrier of preclinical efficacy? I ask that because we, in my institute, largely fund preclinical research, and I don't know what to tell people to look at. So I don't want to put this all on Weiss. I knew I could pick on him with that first question.
Maybe I can comment on just very, very briefly. I mean, my whole life, my whole career has been spent actually developing preclinical models. And I do believe in preclinical models showing efficacy. I do believe that the models must actually resemble the human condition. And so this is why, you know, I've spent my whole career actually developing and studying the contusion model, which I believe is the closest model to human spinal cord injury. And there are lots of things that work in a transection model, hemisection model, that's never worked in a contusion model, and yet some of those things have gone into clinical trial. So you know, there's a lot of give and take on here, and I do want to say that right now in spinal cord injury, we're actually much, much better off than a lot of other fields. You know, for example, like head injury. I've had so many head injury people, scientists and clinicians, come to me and say, we don't have a single therapy that has ever been shown to be effective from brain injury models and applied to human. Now, I've been really shot at over the years for methylprednisolone, you know, which we showed was effective in animals, and then we took to clinical trial and we showed was effective clinically, and then it was very interesting. The clinicians turned around with no data, absolutely zero data whatsoever, just anecdotal data or retrospective data, turn around and say that you know, phase 3 randomized clinical trials are invalid. You know, this really, I've never seen anything like this happen in cancer or any other field.
Actually, in the packet that we passed out, I found some letters to the editor about that are in the packets that everybody has today.
Sure.
So I actually had two.
Can I? The only point I wanted to make is that nowhere in your presentation did you make a single comment about what your anticipated therapeutic target was. That's what I would have liked to have seen.
People getting better.
People getting better, but why?
That's a therapeutic target, Scott. I mean, it gets back to the question of how much mechanism does one need if people are getting better.
I was describing the China Spinal Cord Injury Network, but not to justify the therapy. Okay. I was talking about the network. I mean, if you want to, we can spend the time to talk about it. There's a lot of science behind that. Please don't misunderstand me. I don't believe that basic science should have no part in this. On the other hand, I do want to say something that is really critical, and that is a therapy need not be effective preclinically to be tested in clinical trial. Clinical trials are not to test basic science therapies necessarily. Clinical trials are to optimize clinical practice. That's what clinical trials are supposed to do.
Yeah.
And you're looking at it from the perspective of basic science, but they have another purpose besides doing that.
So I'm going to draw back a little bit because I don't want this to become Weiss against the world. But But we have some people here whose main interest is in stem cells. So having heard a lot more about spinal cord injury than you've probably heard before, and I'm going to throw this to some of the bioengineers as well, who might not have thought so much, but can anyone who comes from more of a very basic background tell me or discuss what they think has come out of this that changes their idea of how their stem cell research should be directed due to clinical considerations? And Scott did a little bit of that, so I thank him, but I was just wondering if anyone else in the audience from that side of the field has any reactions.
Well, I related my concerns with the Parkinson's, and so I had some inkling of what the problems on the clinical side. But you know, I do see that, and I was part of the SMA project as well. That was trying to go from medicinal chemistry to clinical trial, and all through that, I think is very clear. If you know the mechanism or how the disease manifests itself, you can have a much better approach at trying to cure or treat that disease. So the more we know about the mechanisms of the signaling effects and how you might go in and affect those, you can get to a better clinical outcome faster.
Because you can develop drugs that aim at that?
Yes.
Okay.
So you can more, rather than a shotgun approach, you have a more directed approach to that problem. And it's not going to work out all the time, obviously. I mean, I think the biggest problem is again, good preclinical models for these areas, and I'm trying to think of better ways to get better go after areas that have good preclinical models.
So do you think that as basic scientists we first of all need to define exactly what part of spinal cord injury your research is going to focus on? In other words, one of the things that Naomi pointed out in her talk was that getting up out of the wheelchair and walking may not be the patient's first hope. So if we knew that this patient was, what do I mean?
I'll respond to that a little bit. But one of the points that came up in the last discussion was neurological, outcomes that measure neurological function, as opposed to, as Keith implied, functional compensation or other circuits that may not have, not so much repairing the lost circuits as helping the spinal cord as a whole rewire. It's a very big deal in stroke research and brain injury as well. So in the end, I think there is a sort of a grab bag jackpot to all of this, where you are going to do something that's going to have multiple effects. And I would personally say that the real bang for the buck would be in doing enough measures, either preclinically or in early clinical trials, to try and get at what you think is happening. And then in the same ways you do with drug discovery, try and enhance that as you move forward, rather than saying you have to drill down completely before you start a trial. I think you have to accept the fact that there are going to be sort of multiple targets for some of these, and try and be as smart as possible about figuring it out as you go along to make it work as well as it might be enough, to get you to that phase 3 trial.
But let's come up with a couple of thoughts under 2 here. What are some real basic science unknowns?
I think the biggest problem is that you've got cells and molecules and pathways, and then you have behavior. And the closest approximation for a cause for that behavior that's related to those cells and molecules is perhaps anatomy. But we really have this big black box of physiology, of how that anatomy carries out its function, or how those cells and molecules affect either changes in anatomy or how that anatomy functions. I think that one of the biggest holes in spinal cord injury is a lack of understanding how the spinal cord functions when injured and when treated with these things. So
you need an artificial spinal cord.
Well, I think that emphasis on not drilling down to which receptor subtype, but drilling up to, okay, so I put in this stem cell, which makes these factors, which changes these features, and then taking it to the next level and say, how exactly is what I did causing that animal to walk better? I mean, Hans put it up there. Is it myelination? Is it the neurotrophic good humors put out by stem cells? You know, what is the cause? Do you have to put a cell in to get that benefit? So really knowing what's necessary and sufficient between what you've done and what the outcome was.
Can I suggest that this is partially answered by having the gold standard of animal models, and that is to have functional recovery in the animals, right?
But if you don't know what part of what you put in resulted in that, or how it resulted in that.
But if, for example, you found regeneration and an animal did not walk, I mean, this would not be sufficient to take it to trial, right? If you found that you remyelinated and the animal did not walk, then you would not. So at the very least, that there's a more or less some agreement within clinicians that functional recovery is the gold standard in preclinical trials.
But if the cells that you're going to put in can turn into a teratoma, and the reason the animal walks better is because they make factor X, and the right answer is to put factor X in instead of the cells.
Of course.
Yeah, so that's where my point is that if we understood how you got from your cell or your molecule to that functional recovery.
There's absolutely no disagreement, and I think Dr. Stice said it also, that if we understand what the mechanism is, we can actually hone down on a better therapy than the one that actually worked, right?
So is the animal model the best for that? Or should we be getting Steve to make spinal cords in a dish?
They're not the same.
The in vivo environment is such a hostile and different environment. It just
another way to look at it is
well, we've got
don't.
engineers here that can make things in dishes and.
Well, the other thing is don't not study spinal cord injury. I would argue make the case that if you really wanted an easier model for neural repair with a stem cell, pick a disease process where you lose one cell population of one type. Parkinson's disease would be a great example. You get
We're talking about spinal cord.
I realize that, but what I'm saying is that we don't know what to replace or what to fix because it's a big black box. And so...
No, the spinal cord is definitely not a big black box. We know what is necessary. We know the obstacles. We know what regeneration is all about. We know what molecules are stopping it. We know a lot about what stem cells are doing when we put them in. Most of the time, the stem cells don't do anything when you put them in. They just sit there, and they're producing some kind of factor. Many of these factors are the ones that seem to be growth factors. So it's not. I think it's being unfair to characterize the preclinical spinal cord injury work as a big black box. We know a lot about what it does. On the other hand, I think that ultimately, still, the gold standard for preclinical studies must be functional outcome. Would you not agree? That I think most of the people in our field would agree that you would want a functional outcome as a standard for
so
taking it.
So what do we do with the fact that so many therapies routinely give a 2-point change on a BBB scale in the middle of the scale? Who knows why? They all supposedly work with different mechanisms. I'm not directing this at you, but I agree actually with what you're saying. But we do, as a field, have an issue with interpreting what something like that means.
And that's the problem with the BBB scale not being the right outcome measure, which we discussed previously, or it's an issue of there's a lot of common ground to get a little bit better, but
so
no home runs.
We do have a lot more, people are routinely using, we didn't really talk about the outcome measures that are used in the models. I did a little bit of introducing people to modeling, but there's a lot more with footfall, a lot more with allodynia and pain measures being done now, a lot more even with autonomic measures being taken. So I would go with Wise on this one. We have good models. I think in many ways, some of what we were saying when Peter was bringing up the questions, these are the things that should. It's not as if there was supposed to be a recipe in there for all trials. It's those are the things that you should be considering as you come up with a trial. In the same way, there are a lot of tools to study spinal cord injury right now. And depending on what therapy you're developing, you have a lot of choices to make. And hopefully make those with some rationalization that you can explain to other people so they understand why that becomes an important point. Physiology, I think, is something less and less people are qualified to do well. There was a real dearth in training of that for a while there, and I think it is extremely important in this field.
Well, it's hard to get physiologists to study spinal cord injury because they say that how the normal cord functions is so hard to figure out anyway. So from the normal spinal cord physiologist perspective, it is a black box.
So, well, it sounds like that we don't have the right animal models then. It sounds like what you're saying, we do have the right animal models.
I mean, I'm not saying the animal models are perfect, but I think
I think we've got good models.
yeah, we've got good models.
We've got good animal
We have a
models.
good model for
So
the
it's
injury.
not-
The question is, do we have the best
what
model
do
to
you
study
think
stem
is
cells?
here?
So the problem with stem cells is that other than myelination, there's no, to get back what Keith said, there's no single cell target. So that's kind of the low-hanging fruit with respect to stem cells. Neuronal replacement is a bear. You saw some very nice work with some of the motor neuron replacement, but in terms of long-track circuitry or interneurons reconnecting, that's really hard.
So what's going to get over that?
And well, the question is whether stem cells, I think the effects on host tissue and the interaction between grafts and the host environment that result in tissue sparing, in initiation of host responses, those are things we don't understand, and those are probably paramount. And we know so much about, I mean, there's the whole lateral and ventral white matter that all of it contributes to locomotor function and is compensatory when other parts of it are lost. And so to ask specifically about stem cells, I mean, I've thought a lot about this for the last 20 years. And it's a really complicated problem because you only have myelin for a cell type. And so it is growth factors. It is suppression of the inflammatory response. It is maybe regeneration.
What about Robbie's question about endogenous stem cells? Are they there and we just don't know how to
They're there,
get
they
them?
don't do much. And we've yet to
Is
figure
there-
out how to do something to do much.
Is that an area that should be focused on, beefed up?
There are some people looking at that. I mean, it's not a neurogenic zone, but there are definitely progenitor cells in the cord that can turn into some of the glial types. That's been studied reasonably, but I think people haven't given up on the idea of exciting endogenous cells to function.
I would like to suggest that maybe stem cells is one of many therapies for spinal cord injury, and it's not the one and only.
Right.
And so, in fact, most of the therapies are not stem cell related, and I think when we use stem cells, it's often not as their pluripotent nature, but they are providing factors or other things for the treatment. And so you know, on the other hand, there is real science involved with the saccharin and NOGO receptors and blockers, and there's quite a lot of stuff on CSPG, chondroitin 6-sulfate proteoglycan work. There's a lot of science in there that says this is why the axons don't regenerate. The axons come to the injury site. We know that they stop there because there's a layer of CSPG there, and they don't like to go across this injury zone, which is bereft of a lot of signals. They just get lost in there. We also know there's a problem with them after they cross over the injury site to keep growing until they meet their target. So some of these problems have been really well studied, and I'm not so sure stem cells really are an essential part of this.
Well, most
this thing.
stem cell biologists are really developmental biologists. So do
Right.
we need to know more about development of the spinal cord? Do we need,
no.
somebody in the back says yes.
Yeah, I work on the stem cells, and I can understand from the perspective of stem cells and coming into the spinal cord injury, or rather, in other words, trying to mimic the physiology that we find in a normal spinal cord would have been doing. And I see it is a very difficult to generate. Number one, each of the cell types that would constitute normally in the spinal cord. Number two, get them put together in exact ratio or whatever the percentage, and to get them functional. So, in other words, two different level of problems I can identify as a stem cell biology. One, from stem cell to generate pure population of differentiated cell, for example, motor neuron in this case, or astrocyte or oligodendrocytes, as Hans was trying to tell. Two, get them functionally work as a unit that would substitute the loss of function caused by damage in the spinal cord. This is the two level of problem in my.
I think I'd just like to add that if we know the mechanism, as Wise was saying just earlier, that there are other ways to deliver the factor X that Keith was alluding to. And cell therapy is not, in the context of this, there's a slight distortion of cell therapy because that's what we've been talking about. But if you want to look at the problem as a whole, the pharmaceutical companies would much rather you take a pill and get better than transplant cells, right? So there's always the choice of trying to find a small molecule, then if you can't, then you find a protein or a peptide, then there is this hierarchy of preference for what we'd like. And I'd just like to add that we not stop at that, again, there is a whole aspect of delivery that I think makes or breaks certain approaches. I mean, the CNTF stuff and Regeneron and things, I think if you're able to locally deliver things and, you would have less toxicity than if you gave something systemically. I mean, and the technologies to do that, I think, would enhance the efficacy. Even methylprednisolone, I think, if you delivered locally, you would do much better than systemically.
Do you agree that a delivery system might help the problem of cells reorganizing themselves?
I do not think that's the impact, but it's a very complex, it's not a single event that we can do as such. We know so little about the cell, how it behaves in any artificial system. That might improve certain things, but absolutely we need to figure out all the list of components and work on exactly how they can be combined together.
How do you propose that we do that?
It's a whole challenge to probably each of us. I really do not know the answer. I mean, we need to know one thing as a stem cell biology. I mean, I am involved in stem cell biology. I can tell only one objective, like how we can generate pure population of 100% pure populations of one cell type that are capable of interacting with other cell types that would have been we can find in a normal organ, or in this case, spinal cord.
Can I just add a little bit to that discussion and just describe two potentially accepted mechanisms by which stem cells may be beneficial in spinal cord injury? Okay? And Hans referred to one of them, and I think Scott referred to the other one. I think the first one is that stem cells produce many factors that actually influence the spinal cord. And some of these include anti-inflammatory factors, some of these include neurotrophins, some of these include survival factors of various sorts. Okay, so this is a factor-based interaction with the host tissues that can either affect the acute spinal cord injury, the subacute spinal cord injury, or stimulate regeneration. Right? This is just a general theoretical framework to hang what stem cells might do. And the anti-inflammatory thing is relatively new because many people are now using stem cells as anti-inflammatory agents, right? So it's an exciting area, it's a novel area. The second one is to produce cells that actually do some things for the spinal cord, like remyelination. Okay? Now, there are at least 5 or 6 cell types that actually remyelinate the spinal cord. I mean, there's olfactory-sheathing glial cells, Schwann cells, even enteric glia cells, and there are oligodendroglial cell precursors, and there are stem cells, right? So there are many cell types, but stem cells represent one of the most promising ways of getting a source of cells that you can transplant to remyelinate this spinal cord. I think one of the big questions is, does remyelination alone produce that much functional recovery? And finally, what is the rationale of using a remyelinating cell for Asia A when you don't have enough axons, right? I mean, these are the kinds of things that we should be discussing, because these are well accepted, known mechanisms of action and stem cells. So if we can just focus on that, it becomes, I think, an easier way to evaluate whether or not we should do the therapies. And also to answer the question that you've been asking, what is the preclinical efficacy that you need in order to take a therapy into clinical trial? I believe that in order to take a therapy into clinical trial, you need to have an acceptable rationale. Now, Scott questioned my rationale for using umbilical cord blood and lithium, which is fine. It's perfectly fine. What we have to do when we decide whether or not a therapy should go into clinical trials, what is an acceptable rationale? So one of the questions that we should be asking is, what is that level of acceptable rationale? Does it have to fit with known mechanisms of action? And I would propose that it does. If
Even
we
though
don't
you
have a
can't
known
point to which
mechanism
one
of
it
action.
is.
Hmm?
Even though you can't.
I would actually disagree with you on that.
Okay. Okay, go ahead.
No, I mean, I think if you can show that it works, I think that's the, you know, if you can see we're at the same level. You show some kind of functional return, then you work backwards and find your mechanism. But you need, the date.
But you said there are some cases where preclinical trials that are negative still should go to clinical trials.
I think preclinical trials need to show functional recovery before it can go. That's what I think.
I agree with you.
Now,
So in the lithium cases, there's enough to justify that?
yeah, there's functional data, but there's questions whether the functional data is sufficient. I mean, that's another discussion, but there is some data. There are 5 independent groups that have reported that umbilical cord blood is effective. We have some internal data which I haven't had a chance to present, but I believe that you need
data is equivocal.
Yes.
So one of the things that needs to happen is there needs to be more studies repeated.
Yes.
Naomi's talking about that, everybody's talking about that. A lot of these studies need to be repeated, right? And the problem with repeating the studies is that it's very difficult to get funding to do this study over.
Where are we competing the replications? I mean, spinal cord is the only neurological disorder that I know of that has money set aside specifically to be doing that.
Yeah.
So what will help in all that is knowing what it is that needs to be replicated. I mean, those centers have scientific boards. So yes, it needs to be done, but no, I don't think it's one of the barriers because we're trying to do what we can about it.
But this is the process of science, right?
Okay.
There's good science, there's bad science, there's moderate, mediocre science, and in the discussion of the process, we decide what convinces us. And there's some, sometimes one study is enough to say, let's go. There's sometimes 5 studies, all of which are mediocre, and you say, I don't believe, right? This is what science is all about. I think our field is actually very critical. Our field in spinal cord injury is very critical of what it takes to get into clinical trial. I think we have a very high activation energy barrier. I mean, look, if you have AIDS, they'll go with less data than we have now, right? But in spinal cord injury, it's quite rigorous.
Well, AIDS is actually a really good example of a disease that was almost entirely patient advocate driven in terms of how much money the NIH was willing to throw at it. So, I mean, I think that's a really good example. For the first time ever, the NIH funded drug discovery. The NIH doesn't fund drug discovery, but they did with HIV disease. Well, I mean, they did with HIV disease for the first time in a very long time.
Yeah, and AIDS was a real orphan condition when it first started.
And it was a real orphan condition when it started. And everything that the NIH threw at it was totally patient advocate driven. Those people sat on the lawn.
And a lot of drugs went into clinical trial without any animal model.
Yes.
There was no animal model.
One of the things that spinal cord injury needs is more patient advocate input. Then the community needs to know that. Now, I think there's a whole bunch of people in wheelchairs in Washington on a regular basis, so maybe that's just not enough here. But, I mean, certainly I'm very familiar with HIV disease, and that's certainly a patient advocate-driven disease.
Well,
So Scott,
I can't generalize.
Scott,
there's
if you think you have a good animal model, and you really would like to answer more basic science questions, what would it take to get that to happen faster?
I am much less optimistic about stem cell therapy in spinal cord injury than I was previously.
Forget stem cell therapy. Just talk about spinal cord injury. What's going to take? What would take it? What would you need to move your work forward faster? Do you need more money? Do you need something else to be discovered?
Sure. No, I mean, I think there's. I think that what's really involved is for spinal cord is for things to be done not in isolation. This is an example where you're getting people together that do different things, and I think from a research point of view, that's what you need. And there has to be feedback from the clinicians that, and the surgeons and the basic scientists and different kinds of basic science. Well, the field is moved forward so infinitesimally over the last decade, I think. I mean, it's better, but there have been no quantum leaps, and I think it really requires, it's going to require something out of the box that's going to come in that can integrate with what's being done to really move it forward. And there just have not been any real major advances. I don't think there's really significant evidence that regeneration has a real big impact on functional recovery.
So do we need the biomedical engineers to just engineer around the injury? Is that what we need?
Before we leave that, I think Scott said something very significant, okay? We've actually have a large number of therapies that have restored function, but no regeneration. We've actually have even had studies that show regeneration that show no recovery.
Absolutely.
So there's actually a mismatch there in this respect. And you know, I completed, by the way, I completed a study that I had a hard time publishing. We showed beautiful remyelination in the spinal cord, and I can't get the thing published. You know, because we didn't get functional recovery in the rats. And we're very honest about it. We said we didn't get functional recovery. Olfactory and sheathing glial cells myelinate everything in sight beautifully. And we did this 3 years ago. I'm still submitting the paper into journals. So it's a very interesting situation that we actually, the morphology, the remyelination, regeneration may not be actually connected to the recovery of function, which is actually a very disturbing.
That must be depending on what function you're defining.
Walking.
Right.
Well, yeah, we don't have that many. Unfortunately, that's one thing that we are lacking in the preclinical field. We don't have all that many good function in the rats. We don't even have a good sensory function test right now for the rat.
So we do need a different animal model.
No, we need better outcome measures.
Better outcome measures. Okay.
And-
Jose?
So part of the objective of the meeting is, I think, to bring or to share with people what things we learn from the rest that actually surprise us for the first time. So what I think, I'll tell you what I learned is that I came here thinking that spinal cord injury was one kind of disease, but I end up, I think now, believing that it's a syndrome, where you have very complicated lesion that, depending on where the lesion takes place, you're going to have different cells that are going to be needed at certain rates and amounts and ratios. And on the other hand, we have cells that can, potentially we can make all kinds of cells, but one at a time perhaps. Maybe we can only make oligodendrocytes at one at a time. And then the worst thing, the kiss of death, is to find out the cost of production and scaling up for the clinical trials for cell therapy. So I'm with Scott that I'm little, my spirits are not that high in terms of stem cells, and we may, if you really want to study, do cool science and see what stem cells can do, well, yeah, keep trying, but if you really want to help people quickly, you may gonna have to be bold, like Wise is trying to be bold with his approach, or look other ways. And one question I have for Wise or Scott, I don't know if you mentioned that, Naomi, is there anything we have learned or we could learn from lower vertebrates or zebrafish or one of those animals that regenerate spinal cord? Is there anything we could eventually learn from them?
Yeah, so we're using frogs instead of mice.
We're looking to replace a regeneration person, and we're looking for a zebrafish developmental biologist. So that's exactly, that's what I meant by being out of the box a little bit, is pulling in somebody from a different.
Zebrafish is a beautiful model for regeneration, and it does regenerate spontaneously, but it, like
They don't
all.
walk. They don't walk.
But I think you need the development. But you do, and I totally believe in developmental models, and but look, let me take the actually opposite viewpoint, because I've now heard pessimistic views from at least two of you concerning stem cells. I happen to believe that stem cells play a very, very important role in spinal cord injury, in the repair of the injury site, and the main reason is that you know, what encourages me is that you know, what is needed to repair the injury site is not a single type of cell. You need multiple kinds of cells. You need endothelial cells, you need astrocytes, you need microglia, actually. I mean, nobody ever thinks of microglia as good, but I think they're important. You need all sorts of cells. And the stem cell is the only one that can actually produce all those cells and actually recreate normal tissue in the area. And
you're being naive. You're being naive, a little bit naive.
Do you really think so?
Yeah.
No, I actually sort of agree with Weiss. I'm not sure that you
I
want to
agree
put
with
in
Jose.
cells that are
I was naive when I came here. I thought this was a slam dunk. I said, well, come on, we have plenty of cells, let's put them in. But I found that the lesions are totally different, and you may need different cells. I didn't mean to be offensive.
No,
what
no,
I'm
no.
saying is, that you're flying at 10,000 feet, and then you have to come down and say, okay, the cells can do all that, but what kind of cells are you going to put in?
Well,
you can't put a cocktail of something.
I've now, you know, I was not a fan of cell transplantation for many years, as some of you may have known. I mean, I didn't start transplanting cells until 5 years ago into the spinal cord, and I was very skeptical that any cell would do anything at the injury site, right? And basically, my view of the spinal cord at that time is it's the Sahara Desert of the central nervous system. I mean, it's bereft of all signals. There are very few proteins around. It's bristling with all these extracellular matrix proteins that just don't like cells to come in. And it's filled with macrophages even. It's not a good place for axons. It's a terrible place. And I was looking at this thing and saying, well, is there some way we can bypass this injury site altogether, or just even remove it and then fill it with some plastic material or whatever? I mean, that was my view. I've now figured that really cells do something to the tissue that makes it different. You know, I've seen cells actually reduce the cysts that form in the spinal injury site. I believe that a lot of these cells provide the growth factors that actually are beneficial.
When you say cells, I mean, we have 220 different cell types. So which one are you talking about?
I've only transplanted 3 or 4 different kinds of cells, and many of these cells each have individual effects that are very interesting. So we're now, for the first time, trying to do it with embryonic stem cells. The main problem is that we don't have embryonic stem cells for the rat. So we have to go, and now we're starting with embryonic stem cells in the mouse. And one of the major problems we've now discovered is that cyclosporin inhibits regeneration in the spinal cord. It's terrible. Cyclosporin shuts off a lot of the responses that we would like for the tissue repair. And we've now found out that this is...
Cyclosporin, there's a literature.
Yes, because it actually acts through the NFAT, the nuclear factor of activated T cells. And it shuts, cyclosporin prevents NFAT activation. And so what happens is that, what we have now found is that it stops all the lithium effects on the spinal cord. And it's very worrisome.
Robbie also has his hand up.
What?
Yeah.
Robbie also has his hand up.
So I just want to counter some of the pessimism here. I think that we've encountered a couple of problems right now in the field that has prevented us from using the appropriate cells in the area. We don't have a very good source of autograft cells, you know, immune compatible cells to transplant even in the animal model. So that's why I've now spent 5 years. We've just developed the first Fisher rat, green Fisher rat, where we actually can get all the fetal cells, but we still have a problem. We cannot get an embryonic stem cell from any rat.
So that's so one of the big barriers is you need a rat ES cell.
Yes. Or else we go to a mouse model.
Robbie, what were you going to say?
So I've been following the cell transplantation literature, and like Wise, I've been skeptical, and I still am. I haven't been converted, I haven't seen the light yet. But if one had to do cell transplantation, I think we do a very poor job of it. We do a very poor job in the spinal cord. We do a very poor job of it for Parkinson's, the myocardial repair. So there's no success for insulin, diabetes. There's no cell transplantation success story that I know of really in the way that we're using it. And I think a lot of it I think has to do with when we transplant cells. Usually the cell source is a big problem. Not autologous. Where the autologous source is not. The survival is a big problem. The phenotype control after transplantation remains a problem, especially when you put it in an injury site that's complicated. the timing of when you put in and how many cells. Is it the depth of cells that is creating signals that is getting a response? Is it the surgery itself and injecting? So a lot of these are not known. I suggest, but many people have said in different ways, I suggest there's a whole bunch of technologies that you could use in terms of co-transplantation, imaging to track cells, ability to image phenotype non-invasively using molecular imaging or other techniques. So there are a whole host of technologies that I think cell transplantation generally could benefit from and could be of particular use in spinal cord therapies as well. That will help us sort some of these issues out. And these technologies exist piecemeal, but maybe an RFA or something to pull together the science and technology of cell transplantation survival. When you think of stem cells and the recipes involved, they're very intricate. I heard of a 42-day protocol to get to things. But the minute you inject them, you lose control, right? There's no control the minute you stick them in. Is there a way to reach in and have some control after you transplant, right? Rather than let the inflammation or the disease just hope for the best kind of thing. So there are a host of technologies I think you could play with that I can see that give you some control over the environment to improve cell transplantation in general.
Wise was talking about the problem with the xenotransplants and cyclosporin, and argues that we don't have the right model then. So, because if we got to be cognizant of the fact that FDA is not going to let you go into clinical trials with human ES cells if you're based on mouse ES cell data.
Right.
So maybe, we're still at,
I think this is a very significant problem, and that is the concept that you can transplant human cells into a rat and use that as your primary criterion for whether or not you're going to go into spinal cord injury clinical trial, human, is I think a little far-fetched, because you cannot do human cells in rats without giving cyclosporin, at least not most of the time. And I'm very concerned that this is going to be a huge limitation on getting things into clinical trial.
So, wait, I have to respond to that because I was asked here by the CBER people to come, but the Cells and Biologics Division are very cognizant of those issues. And if your preclinical efficacy is mouse cells in mouse spinal cord and you have other supporting data, I don't think their bar will be take your human cells, do exactly the same thing in the mouse, even though we know it's a xenograft and cyclosporin. I think they're very amenable to discussion.
I totally agree, but in the rat it's very difficult to study embryonic stem cells because there's no natural rat embryonic stem cell.
You think they'll let you go to clinical trials without testing the cells that you're going to put in the patients?
They will have you do some tests of them, but I don't think that they won't ask you to do something that is frankly impossible to do, which is take a human stem cell, do exactly what you would in a rat, not acknowledging that you're doing a xenograft.
So as long as the cells you put in don't form tumors in a cyclosporin.
They're going to ask for, as Hans described, they're going to ask for a multitude of data, some portion of which will be the efficacy data. They're going to ask you to test the heck out of your human cells in terms of tumorigenicity, no doubt about it. That's
in a cyclosporin.
That's a different.
Treated.
No, separate from your efficacy, survival things. They'll do toxicity and tumorigenicity, separate from the kind of survival, the efficacy experiment Weiss is talking about.
Weiss, there is a group that actually published rat stem cells. They don't go germline, but they do differentiate into different tissues, so you may be able to take advantage of it. But I actually had lunch today. I followed Anne's advice. I had lunch with people who didn't know, and this turned out to be all the residents of the vet school across the street. So I asked them, I said, what you brought you here? And said, well, we see a lot of cases on dogs that get hurt with this disc when it comes out, and I said, well, how many? I mean, is it really common? I said, well, we see 4 or 5 every week. So, and the owners are willing to do anything, and the bar for, I'm not sure it's going to be probably easier to test certain things in dogs instead of humans. We're having the same problem. We need to develop dog stem cells, but Do you know, is a dog, I mean, a possible
That's
alternative?
been done in a couple of times. I can give some examples. Rich Forgans in Indiana has used dogs in clinical tests, literally clinical tests of veterinary animals. And Nick Jeffrey in the United Kingdom has also been doing that in collaboration with Robin Franklin in some studies. And there's, we actually recently funded a supplement. We have a program called Captor, which is meant to generate clinical basic interactions. And the one that I funded on spinal cord injury was Linda Noble had a collaborator, a veterinary collaborator. This also came up at a workshop we had about a month ago on combination therapies in brain injury, which a lot of similar questions are being asked. And boy, they reacted really negatively to the idea of clinical veterinary trials. Few people there had experience with the vets who basically say there's even more restrictions on what you can do in a veterinary pet than in what you can do in a clinical trial. So it's a mixed bag, but it has been done. They're by no means an easy set of trials to run. But I agree there would be great value in it.
I just wanted to make one comment about a lot of the spinal cord grafting studies, and it gets back to needing rat ES cells. And I would say that a good chunk of the grafting studies have not had proper graft controls, and so they really are uninterpretable. And I don't think there's any cell that you put in that wouldn't interact with the host to cause some kind of host reaction. And so I think the problem with ES cells is that you've got to direct them to something. And so if you're just looking for a cell that is going to interact with the host, you could try a number of different non-ES cells and probably be just as successful. I think the beauty of ES cells is that you can direct differentiation, you can put them at various levels, and that's where I get back to what Jose is saying, is that undifferentiated, they're going to find tumors. Differentiated, we don't know where to put them. And so that's really where my issue is with ES cells as a therapy. I'm not getting down on grafting. I think that's important, but the problem with ES cells is we don't know where to put them, how to direct their lineage before we graft them, unless you're doing oligodendrocytes or motor neurons. I mean, you need directed phenotypes. And so that's really the issue with ES cells.
Well, GABA cells, interneurons play into either networks or
you can take a neuron and you could put in LHX3 and drive it towards an interneuron to drive CPG circuitry. Those are definitive questions. Those can be done. But you need to give an ES cell one function and let it do it. And that's, so I agree with you, there's other neuronal...
You need to be able to follow it after you put it in.
And you need an outcome measure that's appropriate for the lineage to which you've restricted it if you're going to make any comments about functional efficacy of that particular cell graft.
And I just want to say that maybe it's not true that we need a pure cell or one type. Maybe we need more than one type of cell that you put in. Like you might want to put in, you know, astrocytic lineage cells or whatever into the spinal cord as well. So, you know, there are lots of this. Let's have pure cells put into the
The Geron trial, you know, it's mostly oligos, but there is this 5% neurons.
Right.
And some of the, as Geron has gone around the country talking to clinicians, I think, including my center, we've talked about that idea. Well, maybe that's the key.
Right.
And from a clinical point of view, maybe naively, we're thinking that's maybe worthwhile.
Right.
That mix.
Why? Keith was going to say something.
Why? Because there is a lack of neurons after spinal cord injury.
Well, I think part of the whole...
And demyelination is not the only part. It's the number is demyelinated, but the actual disease...
Let me answer Scott's question. The reason why is because no cell is alone. All cells have other cells around them. And it's an unnatural state to be a cell to be 99% pure.
Yeah, that's true. That's true.
Is that...
So I think one of the goals initially when stem cells were first discussed was this hope that there would be a certain interpretability competency that these cells would have, that they would go into tissue environment, figure out what's missing, and differentiate into a population of different types of cells appropriate for that, perhaps in a way that they would get cues during development. Intelligent cells. If you want a large number of autologous cells that make a factor, because that's the critical piece, there's great sources from other tissue types, and stemness may have nothing to do with it. And I'm not yet convinced that there's good evidence that stem cells are as smart as we hope they would be in terms of turning into, I mean, I do believe that in demyelinated areas they tend to make more oligodendrocytes, and in non-demyelinated areas they may make more astrocytes. They do read the cues in the environment, but I'm not sure that they figured out, okay, 15% of you go make, you know, inhibitory interneurons that are inhibitory, and the other ones make interneurons that are excitatory, and you guys make oligodendroglia, and you guys make...
We know they do that. We know they do that in nature. I mean, we know that that's how you make
They
babies.
don't do it in the android spinal cord.
And we don't know how they do it developmentally to get all that complement, to make that complicated anatomy.
We need to wrap up, and this has been really fun, and we can continue this over dinner. I'm hoping that everybody in this room is going to stay for dinner. But that's right. There is a glass. You can have a glass of wine. But it sounds to me like one of the barriers is there is some limitation to animal models. Right? That seems to me like a development of an animal model. I mean, even if you had a rat ES cell, there aren't very many inbred rats. So unless you were making an ES cell from that animal, you're still going to have some issues, right?
Yeah, we actually, Scott might be interested, we actually now developed a Fisher rat, a green Fisher rat that's transplantable. It's really beautiful.
There's an HPAP rat.
Yes, I know. And,
so there's some, but anyway, you don't have an ES cell for it.
Yes, but there's, as Jose says, there's an ES-like cell that's come from germ cells. But I think that the xenotransplant concept is a barrier. I think it's a real significant barrier.
Okay.
I think the model is reasonable, but we need to put in cells that are equivalent, and we need to be able to do it without having to put in immune suppressants, calcineurin inhibitors.
Right. And it sounds like that everybody is agreement that we need to know more about spinal cord development.
Yes.
The development of the cord, the nuances and the piece-by-piece development of the cord. And it sounds like there's consensus that we don't know what happens to these cells after we put them in.
Or don't
That
have
the
means
delivery
of controlling
system and where they go.
Well, one thing I haven't heard anything about is endogenous repair mechanisms and their modification.
Well, I think that WISE is hoping the stem cells enhance endogenous repair mechanisms.
I agree, but I think there's a real deficit not only in spinal cord, which I'm not as familiar with as stroke, but the cells that would be summoned from various areas of the body, such as bone marrow or local endogenous cells, what they would normally do in a repair response.
Yeah.
And some of this may be inhibited, obviously, by the severity of the injury.
Yeah.
But if you understand the underlying mechanisms, you could enhance that response instead of putting in a totally foreign embryonic stem cell. Look at cells that are engineered already to go and help repair the injury.
Right, that was the endogenous stem cell.
So Gene Rathel is doing some of that work in terms of, there are a number of people looking at the NG2 positive cells in the cord as a potential endogenous stem cell source. I think, I'd just like to point out that some of Scott's earlier work with the RN33B cells, for instance, I mean, there has been work, and if you're not from, I mean, I don't, okay, I want to say this in a very positive way, not, gee, you didn't know the answer to that. But there has been a lot of work looking at, you know, which zones are neurogenic and what happens. Some of Scott's earlier work looked at the difference between putting cells actually in the site of injury as opposed to near the site of injury as opposed to further away where it's more intact and the instructional cues are there. And I think that's sort of been in the background of some of these, but I think we have some answers to that. It'd be nice to try and draw what we know doesn't work into building a question of what you can do inside of that lesion. I thought some really interesting data we saw today, I believe it was Hans who showed in his cervical injuries, his cells basically blotted out the entire cyst. I'd love to know what was going on in there. If those were, you know, mainly a big, is that a new kind of scar, or is that some kind of circuitry? I think looking at those kinds of questions rather than, there is some work there we could build on, is what I'm trying to say.
Jose, we got.
One thing that came up here and was also my concern, and the only thing you brought up, what is the role of the patient on the decision-making process of to what level of risk you're going to go for. So I think, no for today, no for this conference, maybe next year, it'd be nice to have maybe one or two physicists trying to address that. I don't know how to do it.
I agree. Can I comment briefly on your statement?
Sure.
I think it's really important. One of the, we of course know a lot about the repair response, but the problem is, for 50 years now, that repair response was regarded to be bad, and that is gliosis. Gliosis is the repair response of the brain and the spinal cord to injury. And yet, there are whole groups of people who think that is what's preventing regeneration, and that is what is bad. And it's a beautiful system. The gliosis is what repairs the blood-brain barrier, that rebuilds the endothelial blood vessels. There are angiogenesis factors involved. And we know that a lot of these cells are also stem cells that are in part responsible for rebuilding the injury site. The injury site does change dramatically over time. We know that there's been a blood-brain barrier breakdown. We know that the tissue is just a necrotic mess, and it actually repairs itself. That reparative process has been deemed by science to be bad. And I've long argued that we should not be using the word scar to refer to what is happening in the spinal cord. It's gliosis. To me, as a surgeon, scar means a fibrosis, fibroblast-mediated effect. What happens in the spinal cord itself is actually a repair process. And so right now, you talk to most patients, they'll say, how can you cut the scar out of my spinal cord? I mean, this is actually what the patients think now. It's so widespread, this concept.
But it's interesting that you mention that, because it may very well take, in chronic patients, a re-injury step, a new cascade of inflammatory event, and then a repair strategy. So we may end up cutting out people's gliosis, scars, cysts, whatever, to get that repair.
Thank you very much. We're going to call this, because I'm sure everybody's blood sugar is as low as mine, although I've been working on it all afternoon.